Welcome to another Purdue Lecture Hall series. My name is Tommy Soares. I'm Assistant Director of the Purdue Institute of Inflammation, Immunology, and Infectious Disease. And today I have the pleasure of welcoming Imrul Shahriar from the Department of Chemistry, who works with Dr. Phil Lau. And Imrul, I'm so excited to have you on the show, on the program, because first, I can't wait to hear your story and your journey of how you got here to Purdue and got into Phil Lau's lab. And second, because Dr. Lau, his reputation and the reason why people know about him is because of all the work that he's done in cancer and cancer therapeutics, cancer detection, all that work that he's been doing. But here you come and you're like the secret sauce to all the virus work that he's been doing. You've been doing all this work on influenza, and it's just awesome to have you on the program and tell us a little bit about that. And I want to hear it all. So welcome to the program.
Thank you so much for inviting me. It's a pleasure for me to be a speaker here, and I'm looking forward to it.
Awesome. Well, I guess maybe if you can share your screen, we can get you going with your presentation, and you can take us back. Tell us a little bit about your journey, what brought you to Purdue, and I'm going to turn myself off so that you can take on the spotlight, okay?
Great.
Perfect. Thank you.
Okay, welcome everyone to the Purdue Lecture Hall series. I'm Imrul Shahriar. I'm a PhD candidate in the Lau Research Lab at Department of Chemistry. So today I'm going to share my journey of how I got into Purdue and in Lau Lab, and the work I have been doing here for the last 3 years, and what's next. So just to get started, I'll start. So I was born in and brought up in Bangladesh, that is 8,000 miles away from the US, very far from here. And how actually I got interested in Purdue, or when I was an undergrad and thinking about applying for a PhD. And I was a big fan of TED Talks, so where speakers from different countries, different backgrounds used to talk about different topics. And one day I came across these 2 TED Talks from TEDx Purdue, one from Professor Philip Lau and the other one from Professor Jonathan Wilker. Both were in chemistry, and I was also a chemistry undergrad. And their talks inspired me so much that I saw professors at Purdue are doing such an excellent job in research, in innovation, and the work they're doing is just amazing. And that's the first time I got introduced to Purdue, and I thought about applying for a PhD. So the talks that Tommy arranges, like this Purdue Lecture Hall series and the Pathfinder series, so all these different talks, this can have actually a big impact, and you never know when it is in YouTube or in social media platforms. So who will get encouraged, and maybe it can change the track of their career. So it is my great pleasure to be a speaker here today. So my journey in chemistry started from University of Dhaka. It's one of the leading universities in Bangladesh. So I did my undergrad in chemistry there. And University of Dhaka is very famous for many individuals. And one person that I would like to mention is Professor S.N. Bose. If you have heard about the Bose-Einstein statistics, so Professor S.N. Bose's name was attached to it. So, and the boson particles are named after him. So he was a professor of physics in the University of Dhaka. So such inspiring personalities always motivated me to dream big and to do something big. And when I thought about what actually inspired me in science and scientific research, I could think about my teachers and mentors in my life. So I was very lucky to have great teachers and mentors who inspired me throughout my life, from primary school to even right now in the grad school. So I think myself as like I was a blank canvas, and my teachers gave it a meaning with strokes of brush and inspired me to dream big. So I'm going to just mention a few of those mentors who actually changed how I think and what I have become today. So my first passion for science actually was seeded or started when I met this amazing teacher, amazing personality, Dr. Ishtiaq Islam Khan. So he was a first-year medical student when I met him in 9th grade, and I was a 9th grade kid in the high school. So he used to teach biology in a coaching center, so where I used to go for tuition. And before attending his classes, the whole meaning of science was different to me. It was like, okay, reading textbooks, and understanding some stuffs, and then sitting for exams. But when I attended his class, he gave me a whole new view of science. So he used to teach biology, and he helped me to visualize what we are reading in the textbooks. So I understood how important it is to visualize a concept, or it actually depends on the teacher who is teaching. If he can make it visualize, make it feel really in a student's mind, that can actually motivate us students in learning, and that can make him very passionate. So he was the first person who ignited my interest in science, and he is a very successful doctor right now. And the second person who I'm very grateful to is Dr. Mushfiq Nawaz Ahmed, so who is also a medical doctor and a pulmonologist right now. And I met him when he was also a student at the medical school. And I was in 11th grade when I attended his biology classes. And he taught all those advanced topics about biology, about human body, anatomy, physiology, those basic things that we learn in the high school biology in such a nice way that inspired me to think, inspired me in science, and motivated me in science. And then another person who actually motivated me towards something other than science is entrepreneurship, is Mahmoud Hassan Shohag. So he was my physics teacher, and he also is a very successful entrepreneur, and an electrical engineer by profession. So he actually made me understand what education really means, or what's the purpose of education is. So he used to say, education doesn't just mean learning some topics or subjects. It means it's just a way to make you think, to think about the problems, to think about the solutions to those problems. And you have to have an entrepreneurial mindset to tackle those problems that you are facing. So that motivation I got from him, that I need to be excited, I need to take initiatives, I need to collaborate with people, work with others, and have that entrepreneurial mindset to be a successful researcher and to create an impact in science. So I was very lucky to have that teaching from Muhammad Hassan Shohag. And who actually brought me into the formal area of research is Dr. Muhammad Halim. So he is currently an assistant professor at the Kennesaw State University in Atlanta, Georgia. But when I met him in my undergrad, so he was a PhD student in CNRS in France. And so his initiative motivated me very much towards research, because he introduced me to the world of computational chemistry. So his master's was in computational chemistry, And computational chemistry was not a very popular subject in Bangladesh at that time. And we didn't have any course on computational chemistry. So I never realized that we can visualize the molecules, the atomic orbitals, molecular orbitals, how electrons are organized around a molecule, the electrostatic potential surfaces, polarity. So all those molecular properties that I have been learning as a chemistry undergrad. So I can actually calculate those and visualize those using quantum chemical tools. And even we can simulate what is happening inside our body, like a protein moving and with water molecules all around it, with drug molecules around it. So we can do a molecular dynamics simulation. So that thing actually excited me so much as I was then a second year undergrad student. So he was the person who introduced me to that realm of research that we can do. And he actually, when he was a PhD student, he opened a private research institute in Bangladesh. And he just rented a small room on a rooftop of a 8-story building. And there was no lift. We used to climb the stairs up to 8th floor. And there was only 2 computers in the beginning. And he used to teach online because he was in France that time. And I'm very, very, very fortunate to have a mentor like him. And he is currently a very successful faculty at the Kennesaw State University. So that's how I got inspired in science through my teachers and great mentors who actually made me understand that I need to actually visualize things, what I have been learning. And I have to have an entrepreneurial mindset to take initiatives, to try out new things, to explore, to collaborate with others. And all those qualities that I can currently have in myself were actually from these great teachers. So moving to next. And when I was an undergrad, actually, because of these great mentors, I was very active in not only in learning everything by myself, but also spreading the joy of science and to inspire the new generation. Because in rural areas at that time, actually, the number of students studying science in high school was decreasing. So there was an initiative, Bangladesh Science Outreach, that was founded to inspire students to study science in high school. So in our country, we had to choose between science, business studies, or arts when we were in 9th grade. So the number of students who were choosing science was decreasing at that time. So this initiative actually was to inspire them. So I got involved and as a mentor of the chemistry and biology team, and we used to design workshops, so 1 or 2 hour long workshops. So that explains for some interesting experiments they can do with their with little things, with even the chemistry experiments they can do with kitchen items, and explaining the complex topics in biology, for example, how blood circulates through our body, and making it very, very simple so that the very young students can understand how science works and how beautiful science is. So it was a great experience to share that joy and love for science with the students. And also I co-founded a small organization, Project Chemistry. So when I was a chemistry undergrad, so I thought how we can actually make a connection between the experts and the new generation of chemists who were interested in doing research in chemistry. So we used to arrange public lectures, like very similar to this one, where we invited speakers from who are very motivating, who are very successful in their field, very dynamic, who can engage with the students. And it had a very great impact. Even there are examples of students who wanted to study engineering or wanted to go to a medical school, but after attending the talks in our chem talk, we used to name it. So after attending chem talk, they got inspired in chemistry and they actually pursued a career in chemistry. So there are examples of that also. So it was a great experience to be so active with spreading the joy of science with the new generation. And what Purdue Lecture Hall series is doing is exactly very similar. And that's why I'm very excited to be here today. So my PhD journey began in the Lau Lab at Purdue. So Professor Philip Lau, who is the presidential scholar for drug discovery, very popular in this field of drug discovery, and a great mentor, a great teacher. So throughout my life, I actually feel very lucky that even in my PhD, I got a mentor like him. So what motivated me mostly about Lau Lab is what I'm doing in the lab today might become a life-saving drug in the clinic. And I can clearly see that path. So there are not many labs in the all over the world that can actually have that opportunity where you can see a drug going from the bench top to the clinic, and you can see the whole pathway, you can experience the whole path. That's something very amazing. And that's something I actually wanted to learn, because when I was introduced to computational chemistry and computer-aided drug design, so we could do molecular docking and simulation in computers. But I always wondered, but I did not have the chance to do experiments or do animal studies or something like that. So I always wondered how actually a drug that is designed using a computer, can that be translated into humans? Can that be how the process looks like, how it actually comes to the market? So I always was very curious about that. And when I joined Lau Lab, that make that concept very clear, very fresh. And as all of you know, maybe that Lau Lab has very successful drugs already in the clinic that are FDA approved. Two examples I want to give, at least one is for the lung and ovarian cancer surgery. So with this drug, they can specifically light up only the cancer cells, and make it easier for the surgeons to do a more efficient surgery and to cut out those cancer cells. And the targeted radiotherapy. So if you just inject a radiotherapy in a non-targeted form, it can be very toxic because it will go everywhere in the body. But in a targeted form, it will just go to the cancer cells and nowhere else. So that drug has also been approved by FDA. So in our lab, we do many different kind of research, working on many different types of disease. And it's a very big lab with 30 lab members, and who are working on variety of projects from cancer, infectious disease, autoimmune disease, inflammatory disease, and so on and so forth. So Dr. Lau is just full of ideas, and the way he pursues those ideas is actually fascinates me very much. So he has some famous quotes. So one of the famous quotes I want to share is, which is actually written in our meeting room in our lab. So he says, one day in the library or one good idea is better than spending 1,000 days in the lab. One great idea can change your career forever. So this quote inspires me so much. And Dr. Lau always inspires to come up with new ideas. And he says, whenever you read a new paper or learn about something, attend a lecture, don't just do it for the sake of doing. Don't just read for the sake of reading. After finishing the reading, close your eyes and think about it, how we can use this information, how I can use this information in my own research. So that's something inspired me to and trained me actually to come up with new ideas, how I can design new experiments, how I can make myself updated to the current state of the art technologies, and to be brave to explore those. So he is a great mentor who actually inspires me always. So he directed me towards the research that I'm doing currently on influenza infections. So the goal of my PhD research is to develop a new therapy for the treatment of influenza infections. And influenza, when I was a student in Bangladesh, influenza was not a big problem. We didn't hear about it much. But after coming to here, I see a different scenario. Here, people are very aware of flu shots, taking flu shots, and the influenza actually is a very big problem and creates a huge disease burden in the US. So as you can see in the last 10 years, from 2010 to 2020, there have been millions of people infected every year with flu, and hundreds and thousands of hospitalizations, thousands of deaths. Even the most recent flu season that from October 1st to March 18th, the very recent data from CDC, they estimate that there have been 26 to 52 million flu illnesses and 12 to 25 million medical visits, hundreds and thousands of hospitalizations, and the number of deaths was also significant, 18 to 56,000. So even though the percentage of death is not great, or most people actually get cured naturally by the immune system, but there's still a huge number of people who requires medical care and hospitalization, and even some of them die. So these numbers are more significant if we think about that currently we have very good vaccines, flu shots, we have antiviral drugs approved by the FDA, but still, this is a big problem. So what are actually the gaps in the field? What we can do to change this scenario, to improve the lives of people suffering from flu? So why do we need a new therapy for the treatment of influenza? So if we look at the current vaccination and antivirals and their limitations, that picture will become more clear. So the vaccinations are great for preventing a pandemic or limiting the epidemic for seasonal influenza, but the vaccination coverage is still very low, 58.6%, and the vaccine effectiveness widely varies from 19 to 60%. With the most recent flu season, 21 to 22 flu season, the effectiveness was only 36% according to the Center for Disease Control, CDC. So we see that we need better vaccines, we need more effective vaccines. It's still not sufficient. And in terms of antivirals, there are many FDA-approved antivirals, but they are of limited efficacy. They only reduce the symptoms by 1 or 2 days, and the therapeutic window is very narrow. So to work them the best, they has to be taken within 48 hours of symptoms. So after you get infected severely, or if you are taking the medications at a late stage of infection, that may not work very well. So these are the current limitations in the field. So if we can design a new drug or new therapy that can reduce the symptoms rapidly, or lower the viral titer rapidly, or extend the efficacy window so that we can treat people who are severely infected with influenza and even at a later stage of the infection. So that can be a solution that can change the current scenario of flu. So before going into all these, as I am aware that a lot of high school and undergrad students might be watching this video, so I wanted to introduce them to the basics of how actually the influenza virus infects our cells in our body. So influenza virus is an enveloped virus. So it has enveloped proteins called neuraminidase and hemagglutinin. So using this hemagglutinin, they bind to a molecule that is expressed on the cell surface in our body. So for example, in our lungs. So those are called sialic acids. So those sialic acid molecules can bind to the hemagglutinin. And after this binding, the virus can enter into the host cell. And the virus is very smart. It can hijack your cellular machinery and produce its own protein and make millions of copies of its own. So it will use your own machinery in your body to make copies of itself and to infect the neighboring cells and ultimately causing a severe disease. So after doing this replication and producing millions of copies, so the newly produced virus particles starts budding from the host cells, and the protein neuraminidase that is also expressed on the virus particles that helps in cleaving off the interaction between sialic acid and these newly formed virus particles, and so that the virus can be released to infect the other cells. So this is how influenza virus infects the host cells and how it spreads. So if we look at it carefully and look at it from the perspective of how our lab thinks about coming up with new treatment strategies, we always want to target diseased cells, for example, the cancer cells, or in case of a virus infection, we want to treat only the infected cells, not the healthy cells, leaving the healthy cells intact. So how can we do that actually? So if we just look at here, so during this viral budding process, the envelope proteins are expressed on the cell surface. So you can see the hemagglutinin, the neuraminidase, these proteins are expressed on the cell surface because the virus assembles on the plasma membrane before budding out. And you will see a lot of these proteins expressed on the infected cells. So that is something not present in healthy cells. So we can use them to target the infected cells. And it would be even better if we can target both the virus particles and the infected cells. So it can be more efficient. We can kill the virus particles and the infected cells together instantly. So for that, we need a protein that we want to target. And we choose a protein, neuraminidase, which is very popular. And there are currently FDA-approved antivirals for that are actually neuraminidase inhibitors. So the good thing is if a molecule binds to this neuraminidase protein, it can inhibit the virion release. So it can inhibit the viral budding process and limit the infection. But still, that is not enough. So how can we make that more powerful? So as I said, we choose zanamivir as our targeting ligand so that it can bind to the neuraminidase that is expressed on the infected cell surface and also on the virus particles. And if you look at the structure, zanamivir looks very similar to sialic acid. So sialic acid is the natural substrate, which is necessary for the virus to enter into the host cells. So here you can see a very interesting drug design strategy. So they made zanamivir look like very similar to sialic acid, and it binds at the same pocket in the protein. And the binding pose is also very similar. So the good thing about being similar to sialic acid is necessary for the virus to survive, to infect new cells. So if the virus wants to mutate or wants to develop a resistance against zanamivir, the drug with which you are treating, it will lose some potency. It will lose its, we can say, virulence, because if it mutates any of these critical residues in the binding site, it will also lose its binding to the natural substrate sialic acid. And actually, we can see that in real. In real, we know that zanamivir is least likely to develop resistance in influenza compared to the other antivirals. For example, oseltamivir. Oseltamivir is more likely to develop a resistance than zanamivir. So it looked very promising that zanamivir has a very low chance of developing a resistance. So we can use that as a targeting ligand. But how can we make it more powerful? We don't not only just want to inhibit the viral budding, we want to rapidly reduce the viral titer. So we want to kill the virus infected cells and the virus particles. How can we do that very efficiently? So there comes the interplay of our immune system. So as many of you may be very may not be very familiar with the immune system. So just a very basics about the immune system. So when our body is exposed to any pathogens, for example, a virus, so there are two different set of armies in our body that can tackle that infection. One is the innate immune system, and the other is the adaptive immune system. So the innate immune system, they are readily available. They can immediately be in action whenever you have an infection. So just to introduce you to the names, the natural killer cells, from the name you can guess it kills. So it has some cytotoxic particles inside it that can actually kill a virus infected cell. And there are macrophages, there are dendritic cells, neutrophils. So they are the first line of responders. So they are readily available. But next, what actually is more efficacious or is more powerful is your adaptive immune system. So those comprises of B cells and T cells. So the B cells, from B cells you get antibodies, and from T cells you get the killer cells and the helper T cells. So the problem with this adaptive immune system, even though it is more powerful, it's very powerful, but the problem is it takes time to develop. So after you get infected, you won't get these cells in action immediately. It will take time. So what can we actually do to harness the power of this immune system? So we have all these wonderful tools in our body. How can we utilize them? How can we guide them to tackle the virus infection? So continuing to that point, so what happens when you have antibodies generated in your body? So when you have antibodies generated against a particular virus, for example, an influenza virus, it can do multiple functions. It can inhibit the viral entry. It can inhibit viral budding, release from the host cell. Also, it can engage the immune effector cells through these FC receptors, and can induce killing of the virus infected cells and also the virus particles. It can also engage the complement pathway, that is another pathway in the innate immune system. So those antibodies can do a great job in rapidly reducing the viral titer and taking care of the infection. But the problem is, when you are first time exposed to the influenza virus, particular strain of the influenza virus, it will take time to develop those antibodies. And influenza infection is a very acute infection. So within a week, it does its job. So if after a week, the adaptive immune system comes into play, they will not be of much benefit. So what we can actually do to achieve something similar or achieve the same efficacy with endogenous antibodies are in humans that are naturally present in all humans. There are antibodies. Can we recruit them with small molecules? So from there, we got our inspiration that what anti-flu antibody can do, can we do that alternatively with a small molecule? So here now I'm going to explain how our drug actually works. So consider what I have explained, how our host cells get infected with the virus particles, and how during the budding process, the neuraminidase proteins are expressed. So now what we are doing is we have designed a bispecific adapter molecule. So it's a small molecule. It's not an antibody, not a big molecule like antibody. It's a small organic molecule. And it binds to the neuraminidase proteins expressed on the infected cell surface. And what it will do, it will inhibit the viral budding. So we are getting what we expect to get from the neuraminidase inhibitors. But there's another component attached to it, actually two components. And those components recruit two different antibodies, anti-DNP and anti-rhamnose antibodies. So these are naturally occurring antibodies present in all humans, although the titer might vary between individuals, but they are present in all humans. So you might be thinking, how do we get those antibodies? So anti-DNP antibodies we get from environmental exposure, because dinitrophenyl compounds are used in pesticides, in wood preservatives. So from an environmental exposure, humans develop anti-DNP antibodies. And anti-rhamnose antibodies, how do we get that? The bacterial cell wall has rhamnose. So humans are naturally exposed to bacteria. So humans naturally have high titer of anti-rhamnose antibodies. So using this bispecific adapter molecule, we can not only bind to the infected cells, but also recruit the endogenous antibodies present in humans. And what these antibodies can do, they can do the magic. They can recruit or engage the immune cells, for example, a natural killer cell around the virus particles, and which can kill the virus infected cells. And the same mechanism can work in case of a virus particle. So that's how we call it a dual mechanism of action. So we are not only inhibiting viral budding, but also engaging with the immune cells to rapidly kill the virus infected cells and the virus particles. So here's just a summary of that mechanism I just explained. So we are using a small molecule adapter and recruiting human endogenous antibodies, anti-DNP and anti-rhamnose antibodies. So that helps us to engage the immune system, the natural killer cells, macrophages, the complement system, to induce the effector functions, so the killing functions, so that the virus and the virus infected cells can be killed immediately. And it's a dual mechanism of action. So now how after, so this is a hypothesis, and let's think about how science works. So you have got a new idea, new hypothesis. Now you need to test it. So how we are going to test it? So first, of course, we have to make this molecule that we are proposing, and it has to be characterized that this is the exact molecule we want. And after this molecule is made, then we need to do some experiments. We need to go to the lab and do some experiments and see whether this molecule actually binds to neuraminidase. Why? Because this molecule, which was originally a zanamivir molecule, has been modified. So whether this modification has affected the binding, we need to check that. So the first thing we do is we do a binding study. So we test whether our drug conjugate, which is a modified form of zanamivir, actually still binds to the neuraminidase protein. And we see, yes, it binds, but with a lower affinity than zanamivir, but which is normal, not very low, still 9.9 nanomolar. So it's still very potent. And so when we are designing this molecule, we have to actually think about that. So think about or design it in a way so that we don't lose much of this affinity. So how can we make sure of that? By looking at the crystal structure, how the molecule actually binds to the protein. And we need to choose a site very we need to actually make an educated guess from this crystal structure for a site where we want to modify, because any modification can lead to a change in the binding affinity or binding of the original molecule. So that's the first kind of challenge we solve. So we find a binding site that is solvent exposed, and then conjugate our payloads like DNP and rhamnose here into that site. So, okay, we verified that it binds with high affinity, 9.9 nanomolar, which is fine. Then we want to test in vitro in cells whether the hypothesis we were thinking about is actually working or not. So we tested ADCC and CDC that I just showed in earlier slide, the antibody-dependent cellular cytotoxicity and the complement-dependent cytotoxicity. So in vitro, we see in this functional assay, it is highly potent with an EC50 in the picomolar range. And we can see 100% killing of the virus-infected cells when we add our drug. And also it can recruit the complement system and induce killing. So this in vitro study tells us that actually, in cells in vitro, we know that our drug is working. Now, the most amazing data will come from in vivo in mouse models. So what we do is we infect mice with influenza virus, influenza A H1N1 in this case, in this particular experiment. So we use a very high dose, a lethal dose, 10 LD50 of influenza. And then 2 days after infection, so when the infection is very much at the peak, we administer our drug. And then 24 hours after administering our drug, we sacrifice some of the mice and collect their lungs and determine how much virus is still left. So if you look at the right in the data, so in the purple, we are showing the control group. So this group of mice were not treated with any drug. So they have a high number of copies of virus, still 10 to the power 6. So 1 million copies of virus still left. So because we haven't treated them. But when we treat with just a single dose of our drug intranasally, with just a single dose, we can completely eliminate the viral titer, the viral load. So 1 million times reduction in viral titer, which was amazing. And we compared with the standard of care drugs that are currently in the market, like Tamiflu and Xofluza, and those reduces the titer by 100-fold to 1,000-fold in 24 hours. But they has to be given within 48 hours of infection. So here we see a drastic effect of engaging the immune system. So if we don't engage the immune system, we will see a lower reduction of viral titer. But if we recruit these endogenous antibodies and get the immune system involved, then we can rapidly reduce the viral titer because they help us to not only inhibit the viral budding, but also to kill the infected cells. Then we looked at the efficacy window. So whether we can treat beyond 48 hours post-infection, which is currently need in the field. So we infected mice with 10 LD50 of infection on day 0, and 4 days after infection, we are treating them with our drug conjugate. And we used intravenous and oral dose in this case, in this experiment. And we can see that the mice that are treated with our drug, as you can see in the green and blue lines, showing 100% survival, even when treated at 4 days post-infection. But if we look at Xofluza and Tamiflu, Xofluza provides 60% survival and Tamiflu provides only 20% survival. So they are not doing very great when treated at later stage of the infection. And also, if you look on the right, we regularly check the body weight. We measure the body weight every day because as a result of virus infection, the mice continues to lose body weight due to several reasons. And we continue to measure the body weight. So if you are treating the mice and clearing out the infection, it will prevent the loss in body weight as well. So what that we see here in case of, for example, when we dose it orally, the EV21, we see no loss in body weight so far in the 2 weeks. With IV, there is some body weight loss, but again, recovers. So compared to the standard of care, it's working pretty well. And then in case of infectious disease, it's very important to have multiple routes of administrations, if possible, for a drug, because some patients who are not very severely infected may want to just take a pill from the over-the-counter medicine, or just go visit a doctor and get a pill and get done with it. But there can be some hospitalized patients and who might require intravenous injection, or which would be more suitable for them. So for that, we tested multiple routes of administration: intranasal, intravenous, oral, and intranasal can also be a great solution for when we are trying to treat infectious disease, because we get infected intranasally, and if we deliver the drug intranasally, it can be very efficacious. So all these 3 formulations we looked at, and we saw that our drug works through all these 3 different routes. We get 100% survival and almost no or little loss in body weight. And then the question comes that you are engaging the immune system. So in case of a viral infection, engaging the immune system can lead to some problems as well. So we can think about inflammation, because these immune cells, they secrete inflammatory cytokines to tackle the infection. And sometimes it becomes overwhelming. So it gives rise to a cytokine release syndrome when the cells are very stimulated due to the virus. So our hypothesis was, if we can quickly kill the virus and virus infected cells, that can reduce the inflammation as well, because the inflammation is caused by the virus. The more virus, the severe the infection, the more will be the inflammation. So if we can tackle that root cause of inflammation, it can also help us uh, getting rid of that cytokine release syndrome, or prevent the cytokine release syndrome. So that's what we have seen. So we have measured the level of different inflammatory cytokines. You may or may not be familiar with this. So these are different inflammatory cytokines that we are measuring. And if you compare with the control group that are not treated with our drug, so we have a much lower level of cytokines compared to the untreated control group. And if we compare to the standard of cares, the Xofluza, of course, does a great job in preventing the inflammation, but compared to Tamiflu, which is actually the most commonly used drug, it doesn't do much greater job for controlling this inflammation. So which is very promising, that we were actually concerned whether it would lead to any toxicity if we engage the immune system, but this data tells us that no, it will actually help. And we also checked the level of different cytokines in the serum, so whether to check for systemic inflammation. So the more the systemic inflammation, the more sick the patient will feel. So if we can prevent that, maybe it will lead to faster recovery of symptoms. So we also saw that for our drug. And then we looked at the histopathology of the lung. So we can really look at the lungs under the microscope after it is stained. And what we can see here, when we don't treat with any virus, so you see this is a histological image of a lung. So if it is a healthy lung, you would see more air space. So the white space you are seeing are the air spaces where actually we have the alveolar air space. So in case of an infection, there will be fluids, there will be those blue things that you are seeing. These are inflammatory cells. So those cells will be increasing in number, and there will be liquids that leads to pneumonia and edema. So all these things we can see in when we don't treat with a drug. But when we treat it with just a single dose of our drug, we can see much better contrast. We can see that the lungs of the mice looks much healthier, much better than what we see in infected mice. Then also to ensure the safety, we also injected our drug in healthy mice to see whether it induces any kind of inflammation or any immune-induced injury. But we see that no, our drug is very safe. When we injected in mice, healthy mice, we don't see any major or any abnormality in the lung and in the kidney. Also, we checked in the liver, in the intestine. So everything looks fine compared to the control group. So our drug by itself is not inducing any damage to these major organs. So that is one important thing to test for any drug. When you are designing any drug, it has to be safe. It should not by itself be a toxic one. And then because we are seeing oral efficacy, so we wanted to test the stability of our drug in simulated gastric fluid. So this is the artificial gastric fluid that has a lower pH like we have in our stomach, pH 1.2, with and without enzymes. And here we can see that our drug is pretty stable, almost 100% stable even after 360 minutes when we tested the last time point. So it's very stable in simulated gastric fluid. It can stay for a long time. It's not breaking apart, which was a good news for us, that it can stay in the stomach for a long time. If we administer it orally, it won't break down at that lower pH. And also we tested the stability in the plasma of different species, because when you want to file investigational new drug with FDA, you have to do some toxicity studies. And for that, you need to choose species, which species you can test the toxicity of your drug in. So for that, the drug has to be stable in that in the plasma of that species. So we injected our drug, and we tested the stability in the plasma of different species: mouse, human, monkey, and dog, and it all looked very promising. So it's not rapidly breaking apart in any of these plasma samples. So ultimately, we came to a solution that meets all the unmet needs in the field. So we have a drug with a novel mechanism, or a dual mechanism of action, and it can extend the therapeutic window. We can treat patients beyond 48 hours of infection, and we can rapidly eradicate the viral load with the help of our immune system. We can administer it through multiple routes. We can develop it as an oral drug, or an intranasal spray, or oral inhaler, whichever seems to be more feasible. And also it provides protection against virus-induced inflammation by eliminating the root cause of that inflammation. And also it provides a higher barrier to drug resistance, because remember, the structure of zanamivir is very similar to sialic acid. So naturally, it is less likely to develop a resistance. So what's next? Ultimately, we want to develop a platform technology for multiple respiratory viruses, not just for influenza. We started off with influenza, but we are expanding to other respiratory viruses, which are very similar, like respiratory syncytial virus, RSV, which is very prominent, was very prominent during the last season, and is a big problem, especially for children. And the coronavirus we are all familiar with. So what we have to do is, it's a platform technology, so we can keep the immunological payload as same. So here we are using a dual hapten. So we can keep the payload as it is, and just change the targeting ligand for each different indications. For example, when we are treating influenza, we are attaching a neuraminidase inhibitor. So when we are going to treat RSV, we can attach a F protein inhibitor. In case of coronavirus, we can attach a spike protein inhibitor. So the same platform technology can be used maybe for a variety of respiratory diseases, and even for solid tumors. So we just need to change the targeting ligand for tumor targeting one. So the same platform technology may be applicable for solid tumors as well. So one good thing about making it a platform technology, and what is very exciting about it, is currently we are facing a kind of a tripidemic. So the coronavirus, RSV, and influenza, all three are circulating together. Many people call it a tripidemic. So to tackle such kind of situation, such a platform technology can be very helpful. So you have a platform technology, you have 3 different syringes with 3 different injections. So you first diagnose which virus you got, and then just give it a shot, and you get cured. So that's what inspires us to build a platform technology. And it was exciting to work here in the Law Lab. And also recently I have been to conferences. So when you are doing science, it's another fun part is you can go to conferences, meet with the experts in the field, and share your knowledge, share your learning, and also learn a lot from them. What I have learned in these 2 conferences I have attended so far in my PhD was incredible. So I just thought, why didn't I came here, attended these conferences before? So one was, these are 2 very prominent conferences for influenza. One is the Options 11 for the control of influenza, organized by the International Society for Influenza and Other Respiratory Viruses, and the World Antiviral Congress. And the one fun fact is in the World Antiviral Congress, the chair of my session was the principal global medical director of Roche, Dr. Aaron Hart. So who is a very prominent person in the field and who actually worked on the development of Xofluza, which is currently the best drug in the market. So when I was speaking about our technology and he was listening to it as the chair of the session, and I got great feedback from him, and also he's a very inspiring and motivating person. So it was a great experience that you are presenting in front of the leaders in your field. So that's something you get the opportunity in science to present in front of a great audience. So I would like to acknowledge the wonderful research lab we have, a very big one with almost 30 lab members. And also the department. Purdue has been a great place for me with so many opportunities. So all the research facilities in Purdue are so helpful. If they were not there, it would not have been possible to build such technologies from an academic lab in Purdue. So I'm very much grateful to all the central facilities in Purdue. So without them, I cannot think of even any of my work. And also the startup company founded by my advisor, Professor Philip Lau, Iradivir. So Iradivir is working to commercialize this platform and working to take it to human clinical trials. And also we are grateful to National Science Foundation for granting us with the NSF SBIR grant and the other investors of Iradivir, so who was always very supportive. And it was a great opportunity for me to work with a startup company along with my PhD. So I was very closely involved and still very closely involved with them. And finally, before ending, some words of wisdom for the young generation of students who are listening to this talk. So what I have learned from my experience in science, first thing is, whatever you read a textbook, you read an article, you read a scientific article from a journal, don't just read it, feel it, try to visualize it, close your eyes and visualize, and think about how you can use this information. That's my first advice. The second thing is be excited about what you learn. It is very important to be excited. So if you're familiar with the first law of photochemistry, what it says light cannot change matter unless it is absorbed by that matter. So light can only change a matter when it is absorbed. So you need to absorb that knowledge or whatever you are learning and be excited. And you need to get an entrepreneurial mindset, take initiatives, collaborate, ask for help. So never be scared to pursue an idea that comes to your mind. So, and another advice that actually I get from my advisor is be brave to pursue the best science, not just what you are comfortable with. So if you can get out of your comfort zone to do the best science, you can be very innovative and be a great researcher. And one very important thing is to embrace failure, because in this journey, scientific journey, you are often encountered with failure. Failure will become a daily fact, but you need to embrace that failures, because what matters is whether you are passionate about it or not. If you are passionate about it, then failure is just another way of learning. So it's just a natural way of learning. So failure teaches you. That's the natural way. So you need to embrace the failure in research. And research gives you the opportunity to mark your footprint in the universe by creating knowledge, solving problems, and bringing science at the doorstep of humans. So what you are doing in the lab can one day improve human health or solve a big problem for the humanity. And you can make a big impact by being a researcher. So all the best for your future endeavors. And thank you so much for having me.
Thank you, Imrul. So awesome. Such a great lecture and so fascinating, all the work that you guys are doing. And the way that you broke it down, a lot of this is like, my gosh, heavy duty experimentation and so on. And you're doing a lot of this work yourself, is that right?
Yeah.
Yeah, I mean, you guys are not only a chemistry lab, but a lot of the experiments and the studies and the trials and the right, the different concentrations and all. I mean, that's all in your hands. That's phenomenal, phenomenal. That's great to hear. And what a journey into a scientific lab that is now allowing you to really move forward in such a great way. You did mention this entrepreneurial mindset to have. It's a mindset, right? It's not like everybody's going to start their own business, their own company, but it's a mindset to be entrepreneurial and say, I can do it. I can take this on, I can learn about this and that and everything else, and we can do this together and find the people that can help you and so on. I really love that. And coming from a lab like Dr. Lau's, of course, it's great. So good. Thank you, Imrul. I will ask you, when you were coming or when you were applying to come to Purdue, like you knew because of these professors, but it's because you looked them up, you did some homework to look what kind of work they were doing in their lab. Is that how you went about it?
Yes, exactly. So first I looked at different universities, and looked at different labs and tried to find out where my interest actually fits. So which work or which lab's work actually excites me.
Yeah,
So yeah, so
that's
that's how
so
I
important. And I love the piece on your mentors. So like so important also to find good mentors, right?
very important.
That's great. That's great. I'm going to ask you a piece of trivia. Let's see if you know a piece of trivia. Do you know what country has the highest per capita rate of vaccinations in the world?
Uh, I'm not 100% sure. Is it US?
No, it's not. It's Bangladesh. And
Really?
do you know which because we know by county, you know which county has the lowest vaccination rates? And
Lowest.
in the world, the lowest vaccination rates in the world are in Marin County, California.
Okay.
And the highest one is in Bangladesh. So there is something to be said there too about how people are coming about information about vaccinations, how people who live also obviously in tropical countries, they have to deal with, rubella, measles, mumps, all of these, but it's incredible to see that difference. We talk about this sometimes in our institute. It's incredible to see. But thank you so much. This has been great, and such a learning experience for me. I hope everybody there also gets to hear it, rewind, hear it again, rewind, and get to see what you guys are doing. It's so exciting. Thank you, Imrul. Thank you very much.
Thank you so much. Yeah,
And have a great afternoon.
you too.
Thanks.